We good? Should I start or
Yeah,
not yet? Yes. Oh,
I
okay.
was wondering if something wrong with the sound.
No.
No.
Okay. Hello, everyone.
Yeah.
Um, Thank you. I want to thank you for the invitation to give this talk, and I want to thank everyone for attending. I know it's very different times in different places in the world, and this is can be challenging, but it's also uh, really fascinating, and it's a great thing that these kinds of things can happen. Um, so these kinds of things are always both an honor and a challenge for those invited to give the talk. And so hopefully I will um, I will meet this challenge well. I provided a somewhat bombastic title to my talk. I mentioned the arrow of time, and I mentioned the high table of evolutionary plant biology. And just to make sure everybody is on the same page, the high table, this idea of the high table comes from the Middle Ages. For those not familiar with it, comes from the Middle Ages where the king or prince or the lord of the domain was sitting at a higher table that was getting better food than the rest of the court or the rest of the attending parties. Um, so this expression, the high table, and here's a more recent one, the British royal family at the high table here, the higher table, better food and all that. So this expression came to mean that being at the high table means you are more important, um, and you are paid attention to. So this is where I was coming from with my bringing paleobotany at the high to the high table of evolutionary plant biology. So,
No, it's fine. I have
so
my.
in 1972, Eldredge and Gould published their paper on punctuated equilibria. And this was at a time when the modern synthesis has basically um, concluded its first phase, and evolutionary theory was dominated by phyletic gradualism. Just to check here, can everyone still hear me?
Yes, we still hear you.
Okay,
It's
okay.
okay.
All right. All
I
right,
just switch off my
yeah.
microphone.
All right, thank you. Yeah, so the idea of punctuated equilibria, along with a few other things and quantitative approaches that were going on in paleobiology at about that time, led to what some have called, or some are calling now, the paleobiological revolution, um, which made it so that by about the early to mid 80s, the field of evolutionary biology and evolutionary theory actually started taking notice of developments in paleobiology. And eventually that led to the paleobiological sciences being seen less as stamp collecting science and being seen more as a significant player, which led to this famous event where John Maynard Smith, famous evolutionary theorist, population geneticist, initiator of game theory in evolutionary biology, was welcoming paleobiology and or paleontology at the high table of evolutionary biology. So at that point in time, in the 1980s, it sounded like everything was great. People were going to pay a lot of attention to developments in paleobiology, paleontology, and paleobotany. However, since the 1980s, um, I think at least that that budding interest and recognition of the importance of the data coming from paleobiology has started to be has slowly been forgotten or started to be neglected. And so this led to things like, and this can be seen most clearly in phylogenetic studies, where oftentimes, except for those studies run by paleobiologists, um, fossils take this auxiliary role where they are used as, let's say, calibration points for dating nodes and things like that. And this has pretty egregious results, as pointed out, for example, in this paper by Wilf and Escapa, who, to keep things short, demonstrated that the ages predicted by molecular clock calibrations with fossils in phylogeny, shown here in blue, tend to be a lot younger, or in general, younger on average, younger than those given by the actual fossils representing those nodes, shown here in yellow. Now, this is not what I'm going to be talking about today. There was a lot of ink has flown about phylogenetics and the role of fossils and the importance of fossils in phylogenetics and so on. But what I want to talk about today is another really, maybe the other really important direction in which the fossil record can contribute to evolutionary biology. And that was also pointed out by Gould, among others, but very prominently by Gould, especially in his book Ontogeny and Phylogeny, where he talks a lot about how fossils inform our understanding of morphological evolution at the organismal level. So aside from the macroevolutionary patterns represented in phylogenies, fossils can inform morphological evolution of organisms. And that's what today we call evo-devo, the evolution of development, or the study of the evolution of development. So what I will focus on is the early Devonian. The early Devonian witnessed the earliest stages of what we call, or what some of us are calling, Devonian explosion. The Devonian explosion as a parallel to the Cambrian explosion, which was also pretty well publicized by Gould, the Cambrian explosion of animal diversity. So, Devonian explosion. Well, some people argue it's not really an explosion. The Devonian, after all, is 30, 40 million years period of time. Nevertheless, whether you call it an explosion or not, what's important to note is that between the late Silurian, in which we already see a significant amount of diversity of very early polysporangiophytes, and the end of the Devonian, basically all the major lineages of plants had appeared, all the way to the seed plants. I'm not talking about angiosperms here, which obviously appeared later, but angiosperms are still seed plants. So if you consider angiosperm seed plants, you can say that by the end of the Devonian, all the major groups of vascular plants had evolved, and actually we see only some of them alive today. Throughout the Devonian, we see the evolution of complex sporophyte organization with stems, leaves, and roots. We see the evolution of the arborescent growth habit, and like I said, the evolution of seed plants, of ovules and seed plants. So a very important period in the evolution of plants. I will discuss some examples of the importance of fossils in understanding the evolution of plant development, based primarily or almost exclusively on plant fossils that are early Devonian, about 400 million years old, from Quebec, from the tip of the Gaspé Peninsula here in Gaspé Bay. That's where we find these fossils, which are permineralized fossils, so they preserve their anatomy. And this is my former student, Kelly Filer, who is very excited to work with these permineralized fossils. And speaking of students, I think instead of leaving it for the end, I'm gonna have my acknowledgements here. These are all students and collaborators who have contributed to the studies that the results of which I'm presenting today. Um, and these are my funding agencies. And I also need to thank the curators and collection managers at the Smithsonian, the National Museum of Natural History, for allowing loans of these fossils that we study in my lab. So I'm gonna discuss 3 examples of the importance of fossils in understanding evolution. These are the 3 examples. We're gonna start with the first one, the sterilization theory and the origin of conducting tissues. And here we're going to talk about these genes called class 3 HD-ZIP genes. That's a mouthful, so I'm just going to refer to them as HD-ZIP genes, but just keep in mind that specifically I'm talking about the HD-ZIP3 genes. These are very important transcription factors that are involved in multiple developmental processes in plants. But what's really important about them, or if you will, the essence of HD-ZIP3 genes, was explored in several papers, and I'm going to bring up 2 of them because they have very important information for the relevant to the evolution of vascular tissues. These papers by Floyd et al. And Prigge and Clark in 2006 that looked at the phylogeny of HD-ZIP genes, they concluded that tracheophytes had a single ancestral HD-ZIP gene, so a single copy that had role in vascular development and is conserved, which is conserved throughout the tracheophytes, throughout the vascular plants. Later on, Vasco et al. In 2016 went deeper in the study of the phylogeny of HD-ZIPs and found that evidence that embryophytes as a whole, so bryophytes and polysporangiophytes and tracheophytes together, also had a single ancestral HD-ZIP, which they proposed, and they provided evidence for it having a conserved role in sporangium development. So on the one hand, if you look at the role of HD-ZIPs in tracheophytes, shown here in blue, it has role in provascular tissue identity, the tissues that develop into vascular tissue, xylem and phloem. But if you look at HD-ZIPs at the level of the entire embryophyte plate, they also have a role in sporogenous tissue identity. This is the tissue inside the sporangia that forms the spores. So it's reproductive tissue, if you will. Now, what's interesting is that both of these roles, based on the studies that I just showed, were fulfilled by a single gene in the ancestors of embryophytes and of tracheophytes. Okay, so what this means is that this gene with double roles implies that vascular and sporogenous tissues share identity, have a shared identity, at least at some level. Now, we can check for that in several ways, aside from doing molecular genetics and molecular manipulations of development and so on. We can also look at that anatomically, and this is where fossils become important, because in fossils, even though we don't see the genes and we can't really follow development, we can look at their anatomy and morphology really well. So if these identities, if these genes, if vascular and sporogenous tissues have shared identity at some level, that means that we should be able to see anatomical continuity between these 2 types of tissues in plants in general, and we should see that the two identities can switch relatively easily from one to the other. So let's see. Actually, if you look in the literature, you find out some interesting things in the old literature. Here's a liverwort. A liverwort, so these are marchantiophytes or hepatophytes, if you will. Porella. In porella, in some specimens of the liverwort porella, the sporogenous tissue, which is found inside sporangia typically, actually replaces the central tissue, the central part of the vegetative tissues of the sporophyte. Liverworts don't have vascular tissues, don't have conducting tissues, but the central tissues of the vegetative parts of sporophytes, that's where we typically find vascular tissues. So this is slight evidence that sporogenous tissue can replace or has shared identity with these vegetative central tissues. On the other hand, looking sort of at the other end of the phylogenetic spectrum, if we look at seed plants, here's Casuarina, an angiosperm, in which Bauer in 1908 showed that sometimes the sporogenous tissue inside the ovules, part of it differentiates as tracheids, as vascular tissue, as xylem cells. So there's this replacement of sporogenous tissue with conducting tissue. So we see basically there are some examples that suggest that the two identities, sporogenous to vascular and vice versa, can switch pretty easily. How about anatomical continuity between the two tissues? Well, if we look at Xylophyton, which is somewhere here in this phylogeny that's based only on living plants, this is what Xylophyton looks like. It has sporangia that are paired. They occur in pairs. And here are some images of the sporangia. Here's the pair of sporangia attached at the base and supplied by the same vascular strand, which branches to supply the two sporangia. And these are cross sections through two sporangia in one of these pairs where we see the sporangial wall and we see the spore mass. So here's a sporangium in Xylophyton. These are serial sections through the same sporangium. And if you zoom in on the base here of the spore sac, you see tracheids here. You can also see them well here that go all the way to the mass of spores. Here's a better example. These are cross sections through a pair of sporangia going from the base here toward the tip where here we reach sporogenous tissue. Now here we see the common vascular bundle. Here it has split. We see two xylem bundles. And if we zoom in on the top sporangium here again, common bundle after the split, xylem. And here we start seeing xylem around some spores. And here a little bit of xylem and spores. And here spores and just spores. So zooming in some more on that series, here we see a lot of tracheids, clearly recognized by their typical Xylophyton type thickenings. And this is the very bottom of that mass of spores. So basically what we're looking at here is sections through this area that shows a continuous, sort of a seamless transition between the vascular tissue and the spore mass. So the central core of tissue in the Xylophyton axis has continuous tissue that switches from vascular to sporogenous, to spores. So basically what that means is that as the axis of Xylophyton were growing, they could keep on growing vegetatively, or a developmental switch was turned on, which made the central tissues differentiate into spore or sporocytes, rather, the cells that will produce sporangia, instead of xylem, instead of vascular tissues. So we see anatomical continuity between vascular and sporogenous tissue. We don't see that just in fossils. Here's an example from Xylodon, a living whisk fern, where we see vascular tissue. Here are two of the sporangia and the synangium. We see vascular tissue here going right up against the content of the sporangia. Here's a detail of that. Here's the trachea going right up against the spore mass. Back to fossils, we don't see that only in early Devonian Xylophyton. This is an ovule of Microspermum. This is Pennsylvania, the Carboniferous seed plant. And if we look here, what we see is the megaspore. This is actually MM is the megaspore membrane. And if we zoom in, we see a vascular bundle with tiny short tracheids that go all the way up to the megaspore membrane. So the xylem is in direct contact with the megaspore there. So there is good evidence for anatomical continuity between the two tissues, vascular and sporogenous tissues, from different lines of evidence from both living and fossil plants. So these support this hypothesis that vascular and sporogenous tissue share identity and regulation. So what I propose is that, again, vascular and sporogenous tissues share identity, at least at the deep level. Obviously, they are not the same tissue, they have different functions, but at a deep identity level, that's shared. That identity, the shared identity, is the identity of the precursor tissue that developmentally forms either one or the other. And I'm going to call that the pre-sporogenous tissue. And that shared identity involves expression and roles of HD-ZIP genes. So this is actually pretty similar to, it's not new in a way, to what F.O. Bauer was proposing in his sterilization theory, where basically he proposed that the vegetative body of the sporophyte arose by sterilization of sporogenous tissue. Now, Bauer did not propose a mechanism for that, and obviously at the time we didn't know much about the genetics of all of these, of the genetic regulation of development, and we didn't know even as much about all the fossils that were discovered afterwards. But now, with what we know, we can start asking questions and refine this general hypothesis of sterilization. So, how can we do that? Well, let's look a little bit at the history of the plants, more specifically, and the genes that are proposed to do things inside the plants. So what we have here is a phylogeny that includes the bryophytes, and here it doesn't matter whether the bryophytes are paraphyletic or they form a monophyletic group as proposed more recently. The bottom line is that there is a common ancestor here of all of the embryophytes, bryophytes plus polysporangiophytes, which include the tracheophytes. So Floyd and Bowman's work, or what Floyd and Bowman proposed, was that based on their work on HD-ZIPs, was that HD-ZIPs have functions in conducting tissue differentiation, and that later on, so they had these functions either in somewhere among the bryophytes or at least in the common ancestor of polysporangiophytes, and that later on the common ancestor of tracheophytes acquired or evolved genes that were downstream targets of HD-ZIPs that were responsible for the actual development of tracheids, secondary cell wall thickening and lignification. Now, what Vasco et al. Proposed based on their study was that HD-ZIPs had a function in sporogenous tissues in the common ancestor of all the embryophytes. So that could normally be explained by a duplication from this one gene that had function in sporogenous tissue in all of the embryophytes to it would, by duplication, a second copy would pick up a function in conducting tissue differentiation. However, we do know, as shown by both of those papers, that there was just a single HD-ZIP in the common ancestor of embryophytes and of polysporangiophytes. So then here's a hypothesis that could explain how this single copy would have been responsible for the evolution of vascular tissues. So remember, pre-sporogenous tissue is this tissue that, in my hypothesis, can differentiate either as sporogenous tissue or as vascular tissue. And so the ancestral function of HD zips was in, if the ancestral function of HD zips was in pre-sporogenous tissue identity, then in the ancestral embryophyte here, which is thought to have consisted pretty much only of a sporangium. It had no big sporophyte body. The whole sporophyte was a sporangium. So there, pre-sporogenous tissue, its identity was defined by this single HD zip, and then that's pre-sporogenous tissue differentiated into sporogenous tissue that formed spores in these reduced sporophytes that were just a sporangium. And so in embryophytes, in the more evolved embryophytes like bryophytes that actually have an elongated sporophyte, the expression of the downstream target of HD zip that determined differentiation into actual sporogenous tissue was delayed by the insertion of apical growth. Apical growth, which led to just elongation of the sporophyte in bryophytes, to elongation and then branching of the sporophyte in protracheophytes and tracheophytes. But eventually, in all of those groups, this downstream target of HD zip, of the one HD zip, would make the tissues at the tip of the axis differentiate into sporogenous tissue. And then, in starting definitely in the protracheophytes and tracheophytes, maybe in the bryophytes, a second downstream target of that still single HD zip gene was responsible for the differentiation of vascular tissues. So basically what I'm proposing here in a nutshell is that the ancestral HD zip defined pre-sporogenous identity, and then two different genes acted downstreams of it to lead that pre-sporogenous tissue to differentiate either as sporogenous tissue, sporangia, or as vascular tissue. And that could explain why we have a single HD zip gene in the common ancestor of all embryophytes. Now, I have a question mark here for the bryophytes because actually, if you look at bryophytes, a lot of them, and these are all sporophytes, but also some gametophytes, have conducting tissues. They are not xylem and phloem, but they are specialized conducting tissues. So this whole thing could have started in the bryophytes, but we don't know until we're going to look into that. So here's the conclusion of this part of the talk. Basically, I'm proposing that vascular and sporogenous tissues share identity and regulation at a fundamental level. This shared identity involves regulation by HD-ZIP3 genes. And this supports a sterilization scenario for the evolution of conducting tissues. This hypothesis that I presented has testable parts, looking at the living plants and developmental regulation in living plants to understand the evolution of developmental regulation of both vascular and sporogenous tissues. And I think these tests should involve bryophytes and looking at the conducting tissues of bryophytes and what leads to their differentiation. That may help a lot because they are so basal in the phylogeny of embryophytes. Okay, second main theme, tempo and mononuclear diversification and euphyllophyte morphology. Here I'm going to take a much broader look. And again, we're looking at the early Devonian, where we see coming from the Silurian, we see these three main groups of early polysporangiophytes, some of which are tracheophytes. We see zostrophils, we see trimerophytes, and rhiniopsids, as well as protracheophytes. Just to remind you, here are rhiniopsids, rhinia. These are zostrophils with their exarch xylem maturation here. And here is xylophyton that we've already seen with its centrarch primary xylem maturation. Now, we also think, and there's pretty good evidence, that the two main extant lineages of vascular plants, the lycophytes and the euphylophytes, arose from one from among the zostrophils and one from among the euphylophytes. But in reality, this is what we know. There's some plants with vascular tissue, with or without vascular tissue, but with branched sporophytes, that we know all the way into the middle Silurian, and from there on. Then, if you look at the level of the Pragian to Amcian or so, and also in the Locovian, I guess, we have zostrophils and trimerophytes, and we have a lot of diversity, but their relationships are not well resolved, really. So we don't know how these earliest polysporangiophytes relate to all of the diversity we see in the early Devonian. And then, by the late Devonian, we see representatives of well-defined lineages, some of which have living relatives, some of which don't, but they are really well-defined and differentiated from each other. But again, we really don't know how these early tracheophytes, zostrophils and trimerophytes especially, are related in a phylogenetic sense to many of these groups. So lack of resolution here, primarily for lack of enough good fossils reconstructed as whole plants. Even so, by looking at early Devonian fossils, we can find out a little bit more about the evolution of morphological diversity in this case of euphylophytes. That's what I'm going to focus on. And so I put this here. This is a symbol of the xylem architecture of Xylophyton. The xylem is in red, and this darker red dot is the protoxylem, which is central in Xylophyton. So let's look now at these other, what we know about these other lineages in terms of their xylem architecture. In the middle to late Devonian, we have Iridopterids with very complex xylem architecture, which I'm going to diagram like this. So I'm going to show these diagrams to sort of summarize just the general pattern of xylem architecture for each of these groups. These are all fossils that have been described a while ago, many of them, so I'm not going to spend too much time on them. But here's Iridopterids, pseudosporocnalians. Just like the Iridopterids are known from the studying in the Javedian primarily, pseudosporocnalians earlier in the Eifelian. There are sphenopsids, known from the Femmenian at least. Progymnosperms, aneurophytes, such as Aneurophyton and Tetraxylopterids, that we know from the Eifelian. Archaeopterid progymnosperms, Archaeopteris and closely related taxa, known from the Givetian. And Stenocolealians, yet another lineage that we don't know for sure how it relates to the others, known also from the Eifelian. And finally, seed plants, we know them from the upper late Devonian. They are probably older. Okay, so this is the summary of our types of xylem architectures that we see in these different groups that we know from the middle and late Devonian, well-defined groups, like I was saying. So if we actually map them in terms of time with the earliest known representatives, designated by the base of the circles, this is where we see the origins, or at least the earliest evidence for each of these xylem architectures. Okay, so how do we get from this very simple xylem architecture that we see in Xylophyton and the other trimerophytes, from here to all of these which we know they are descendants. There's pretty good evidence that they're all descendants of trimerophytes like xylophyte. So let's look at the Battery Point Formation, the fossils from the Battery Point Formation in Quebec that are again about 400 million years old. They are middle to late Emsian in age. So I'm going to show you some fossils, some of which we described, some of which we're in the process of describing, that exemplify again the different types of xylem architectures. So here's a Dolocodoxis, a cladoxylopsid. This is Leptocentroxyla. We don't know exactly what it is, but its xylem architecture is probably like this. Another plant, Jovingera, that has architecture pretty similar to that of seed plants. In some respects, Pistenoloboxyla, Henricia, Gumujij, a plant with secondary growth. Also, this is a new cladoxylopsid that we hope to finalize writing up soon. And this is another plant that we're in the process of publishing that may have an organization like this, like Amyrophyteliid progymnosperms. Sorry, like Archaeopterid progymnosperms. So, just to summarize, what we see in these plants from the Battery Point Formation is these types of xylem architectures, maybe this one, not sure. On top of these Battery Point Formation plants, from rocks of the same age elsewhere, there's this plant described by Gensel in 1984 and Gotanophyton described by Remy and Haas. So if we add their xylem architectures to plants of that age, we have basically the whole range of architectures represented. So what that means is that if you look at middle to late Emsian plants, this entire sequence of xylem of appearance of xylem architecture actually becomes this. Except for what we see in the late Devonian sphenopsids, we see all of these architectures, very likely. Some of them more sure than others, but there's a lot of xylem architectural diversity in the euphylophytes by the late Emsian. Now, going back to Gould, which put forth a lot of interesting ideas, looking at morphological disparity, the diversity of morphologies sort of independent of taxonomy, which applies to these early Devonian plants, which we don't know, again, how they are related to the later Devonian plants. Gould proposed this pattern of early high disparity, proposed that in the history of major clades, what we see is a very early major diversification of morphologies, which is one of the macroevolutionary trends observed in general in clades, others of which include increasing diversity, increasing organismal complexity, and increasing maximal organism size. So early high disparity. It's a pattern that has been confirmed in most major clades of organisms. It has not seen a whole lot of work in plants. Here's this paper by Oyston et al. That actually looked at morphological disparity in several major plant lineages, and they showed that indeed in many of them or most of them, we do see the same pattern of early high disparity in the history of each clade that we see in other major clades of organisms. So what these data from the early Devonian show us is the same idea that we see a very quick transition from simple xylem anatomy to very diverse types of xylem anatomy. So we see early high disparity, at least in terms of xylem architecture. Now, what's interesting is that if you look at the morphology, the external morphology of these plants, they all are simple. They don't have roots. Most of them don't have roots, at least among the euphylophytes. Yeah, they haven't been euphyllofites of this age, they have not been demonstrated unequivocally. They don't have leaves, so they have very simple morphology. They have these simple branching axes, and the evolution of we see the first leaves among euphyllofites only toward the end of the Middle Devonian, after which we see another explosion, if you will, of disparity among euphyllofites, but that's morphological disparity with different types of leaves and the evolution of roots and so on. So the overall morphology of the sporophytes sees an expansion in disparity. So what this means is that, what this tells us is that, yes, we have early high disparity in the euphyllophite clade, but that the evolution of disparity was biphasic. We see a first stage in which anatomical disparity increased very fast, or specifically the architecture of xylem, and then a second phase in which morphological disparity increased rapidly later on. So two phases, biphasic early high disparity. And this summarized is what we just discussed. The evolution of the disparity was biphasic in euphyllofites, first in anatomy and then in morphology. Okay, let me see. We're doing well. The third example has to do with the regulatory toolkit for secondary growth. So we control the developmental regulation of secondary growth, specifically of secondary xylem, because that's what we find preserved well in many fossils, and especially in the very earliest fossils with secondary growth. So I'm going to call that cambial growth as well. Cambial growth is a very complex developmental process, as I'm sure all of you are aware. We'll look at plant anatomy. There's a lot of different patterns of cell division and cell differentiation involved. And this part of the talk is based mainly or almost exclusively on work done by my student Kelly Filer for her master's thesis. So secondary growth, vascular cambial growth that produces secondary xylem, is recognized by several anatomical features that we term anatomical fingerprints. Because like fingerprints, they demonstrate the presence of something. So one of them is radially aligned files of cells that we see. They are radial because of the pattern of cell divisions in the cambium. And we see these radially aligned cells produced by the cambial initials that divide periclinally to the outside, forming secondary phloem, to the inside, forming secondary xylem. So one fingerprint, one structural fingerprint is radially aligned cell files. That allows us to recognize secondary growth. Another fingerprint of secondary growth is anticlinal divisions that lead to the addition of new files. And here's an example here. Here's another one here. Here's another one here. So here, basically at this point, when the cambium was at this point, the cambial initial, the fusiform initial, divided anticlinally to form two fusiform initials, each of which then went on to produce its own file of cells. A third structural fingerprint of vascular cambial growth is the fact that it produces two major tissue systems within the secondary tissues. It produces an axial component, which is the actual conducting cells and other cells that are auxiliary to them, like tracheids and vessel elements in the xylem. So that's the axial system, and then it also forms a radial system, a radial transport system. That's what we call the rays. Okay, so three major structural fingerprints that allow us to recognize secondary growth in plants, living and dead. Looking at the distribution of these different fingerprints and at other features of secondary growth, both anatomical and regulatory, with Andrew Groover, we proposed that vascular cambial growth is regulated in a modular way. So in other words, that vascular cambial growth consists of several processes, each of which is regulated by a separate module, is controlled by a separate regulatory module, set of gene interactions that are relatively independent from each other. And in our 2019 paper, we basically brought together all the evidence from living and fossil plants, from anatomy and regulation, that support this idea of modular regulation of cambial growth. Well, again, looking at early Devonian plants, we can learn more about the evolution of regulation in cambial growth early on in the euphyllophytes. So what I'm going to show here is the oldest four plants that are known with secondary growth, and that have been also described in more detail. There's a few others, but these, we've looked at them in enough detail to be able to compare them. So the plants are Gemugij, Franhuberia, and this plant that I showed before, I'm going to call it plant X. It's not published yet, so we haven't given it a name. All of these come from the Battery Point Formation. And then this is Armorica phyton, which was described by the Gerien and Gensel and Strulu-Derrien team, and with more detailed studies done by Gerien and Gensel in 2016 to describe its anatomy. So these are all early Devonian plants. Like I said, these three come from the Battery Point Formation. Armorica phyton comes from the Chateaupan Formation in France, and it's 5 to 10 million years older than these three. Okay, so let's look. We're going to look at these four plants and look at their anatomy to try to understand the different developmental processes that formed their anatomy the way it is, the anatomy of their secondary xylem, the anatomy of their wood the way it is. And so I'm gonna go through a set of anatomical characters that tell us something about development. So we're gonna build up this table where we have the four plants and we're checking the presence or absence of different characters. So periclinal divisions of cambial initials. These are the divisions that actually form secondary tissues. And they are, the structural fingerprint for that is the fact of having radial files of cells, which we see in all of these plants. All plants have this. That's how we initially recognize them as plants that probably had secondary growth, that probably produced wood. So all of them have radial aligned tracheate files. Synchronization of periclinal initials. So the cambium around the stem, let's say, all the cells in the cambium, typically in living plants, divide at about the same time. So what we see is that all the cells produced by one set of cambial divisions are all aligned, because the cells that the cambial divisions that produce them, they all happen at the same time. The cells were produced at the same time. They grew and differentiated at the same time. So this is what we call synchronized periclinal divisions. All the cells in the cambium divide at the same time. We have nice tangentially continuous layers. We see them in plant one here, plant one here is Gemugij. We see them in Armorica phyton. We see them in Franhuberia. Not so much in plant X. Plant X was lacking them. So the tangentially aligned tracheates are lacking in plant X. Anticlinal divisions of cambial initials. I'm calling them symmetrical because these are the divisions that produce from one file of cells, two files of cells of the same kind. So we see one file of tracheates, two files of tracheates in Gemugij. Same thing in plant X, Armorica phyton, and Franhuberia. So here they are. That's what we call multiplicative divisions. We see them in all four plants. Okay, a radial system. Remember rays, another fingerprint of secondary growth, structural fingerprint of secondary growth. Well, as it turns out, you can produce rays in multiple ways, or at least you can produce a radial system of cells in multiple ways. The typical way in which rays are produced in all living plants and in many extinct plants with secondary growth is by asymmetric anticlinal divisions of cambial initials, where a cambial initial divides to produce one cell that will form the ray and one cell that will keep producing tracheids. Okay, so asymmetric anticlinal divisions, that's what produces the rays. And here we go. So a division in the cambium produces two types of cells. And that's what we see. Those are typical rays that we see in most plants. That's what we see in Armorica phyton and in Frankuberia. Here. There's another way to produce a radial system, and that's what we see in Armorica phyton and Mujic, where basically what happens is the cells produced by the same cambial initial can differentiate into either tracheids or parenchyma. And then, so there's a switch that gets turned on and off to make these cells differentiate either as parenchyma or as tracheids. So that's what we see here. Tracheids, 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 switch on, parenchyma, switch off, tracheids. That's what we see in Mujic. Tracheids, switch off, parenchyma, switch on, tracheids. So we see a radial system formed by asymmetric anticlinal divisions with typical rays in some of the rays in the Armorica phyton and in Frankuberia, but other rays in Armorica phyton are formed by this reversible cell identity switch, like here, and we see the same thing in Mujic. So here we go. We see that Mujic, Armorica phyton, and Frankuberia, they all have a radial system. What about plant X? Plant X is interesting because it doesn't have true rays, but if you look in cross sections, you see that it has tracheids that grow radially, or tracheids, this is a longitudinal section, tracheids whose tips are bent and oriented radially. So that tells us that there is at least there's a radial system formed here by the directionality of cell expansion, by the way the cells grew, although they all differentiated as tracheids and there were no parenchyma, but still there's a radial system. So we have a radial system also in plant X, but if you look at them in detail, the types of radial systems that we see in these four plants are different. We see typical parenchymatous rays in Armorica phyton and Frankuberia. We see parenchyma replacing tracheid files in Mujic and Armorica phyton, and we see just these radially oriented tracheids or tracheid parts in plant X. Now, like I was saying, these anatomical features imply the activity of or certain developmental patterns, which, because these different anatomical features are not always combined, they are not always occur in all the plants, must have been regulated independently, at least to some extent. So here in this table in black, we have the actual anatomical character, and in green we have the developmental process for which this anatomy provides evidence for. So each anatomical character provides evidence for a certain developmental process, and therefore for the presence of a regulatory module, of a separate regulatory module for that process. So what all of this means is that this is pretty strong evidence that there are, first of all, we see multiple distinct modes of secondary growth very early in the evolution of secondary growth. So these are the oldest plants with woody growth that we know. We've seen a mosaic pattern of expression of these different regulatory developmental modules, since they are not all present in all the plants. Different plants have different subsets of them. So what that means is that these early euphylophytes were experimenting within the morphospace of secondary growth by using different combinations of these developmental regulatory modules. So basically, this is strong evidence that the regulatory toolkit for secondary growth has a modular assembly, and that not all the plants with secondary growth have necessarily all the features and all the developmental processes of secondary growth. And this has interesting implications because, for example, we can ask the question, well, what is the minimal set of developmental processes and regulatory modules that you need for a plant to have some form of secondary growth? Things like that. Okay, so in conclusion, we can bring paleobotany to the high table of evolutionary plant biology. And here I showed examples of how fossils can inform macroevolutionary patterns, like the temperate mode and the diversification of phyllophyte morphology with that early high disparity, as well as, strictly speaking, evo-devo questions like the origin of conducting tissues and the origin and early evolution of secondary growth, of woody growth. So biological evolution is the history of life. And this is probably what Steve Gould had in mind when he said good naturalists must be historians. Indeed, Gould recognized that evolution is the science of history and its influence. He said we come to our local environment with the baggage of eons. What he was implying here is that the only way to understand evolution is by reconstructing this history, by following the arrow of time, by basically looking upwards to the geologic eons and following the arrow of time, to use one of Gould's favorite metaphors here. So this is also what Wilson Stewart meant in his upward outlook idea, that the only way to understand evolution and morphological diversity in plants, in Stewart's discussion, is by taking an outlook in which morphological diversity, or the morphology of any given species at any point in time, is the result of a sequence of evolutionary changes of prior forms. So upward outlook, so that understanding a certain morphology can be reached only by looking at what it came from, and all of these changes, and these prior forms are fossils. Stewart was actually echoing earlier views of F.O. Bauer, famously noted that an upward outlook is in itself a practical application of any evolutionary view. Now, Bauer also noted that this seems like a platitude today being 1935, but it is not difficult to quote from current literature instances where it has been neglected. Well, ironically, this is still true today in many cases, and it is up to us, paleobiologists, paleontologists, and paleobotanists, to support and promote and apply this upward outlook. And if I were to summarize this in some recommendations, I would say, how do you do that? Well, look for anatomical and morphological features that can be used as fingerprints, as structural fingerprints for processes of organismal biology, for processes in the organism, such as development, physiology, gene expression patterns. Many of them are reflected in specific phenotypic features that can be recognized in the fossils as well. And once we recognize those, like I showed in some of my examples, we can then compare these structural fingerprints in either an empirical phylogenetic framework or a hypothetical phylogenetic framework, where we can include both plants and fossils, because we can observe anatomy and morphology in both of them. All right, thank you for your attention.
Thank you,
This
Mihai.
is it. Okay, I went over only 3 minutes. Not too bad.
Thank you very much. Very, very interesting.
Yeah,
Thank you.
yeah.
Yeah.
Thank you.
Now everybody welcome to show their faces. It would be much more pleasure to see the faces, not only the family printed. And if anybody want to ask questions, it's right time to do it. Just
I have a
raise your
question for
hand.
you. Yeah.
Uh,
I have a question for you, Mihai. This
Yeah,
is
yeah.
for Roswell. I want to congratulate you on a really nice synthesis of our understanding of structural diversity in relation to paleontology. And more specifically, when you're looking at secondary growth and you're looking at the three parameters of structural features that you identified, when you have only radially aligned cells, how do you distinguish secondary growth, that is paraclinal divisions of a cambium, from radially aligned metaxylem?
Is this a test, Gar?
No, it's a question with which I've been struggling.
Well, I would send you to a paper by, if I remember well, Garfunkel and Rothwell in looking at the radial line tracheids of Botrychium. And they have some pretty good criteria there. Okay, so jokes aside, how do you distinguish it, or what? I guess the way I want to take this question is, where do you trace the line if you only see evidence for some of these separate developmental processes, right?
Right.
And well, so you can use criteria like those used by so other criteria like those used by Garfunkel and Rothwell, who basically looked at in the Botrychium system. Botrychium has relatively easy to recognize annual growth increments in the length of the stem, and so you can follow tracheids, the same trachea in serial sections from one annual growth increment into the next. So, which means that that trachea was around for more than 1 year, and it elongated alongside the stem. It grew alongside the stem. So that's not what typically what tracheids in secondary xylem do. So that's one way, I guess, to look at it. But with fossils, it can be hard to use that criterion. But otherwise, I would argue, and I've encountered opinions saying, well, if you only see radial aligned tracheids, or if you see radial aligned tracheids and multiplicative divisions, so those anticlinal divisions, those are pretty good. Those are, let's say, radial aligned files of tracheids plus multiplicative divisions, to me, are pretty much all you need to argue pretty strongly that you do have secondary growth and you do have wood. And race may or may not be there. And like I was saying, if you take all of these processes down further and further and further down in time, at some point we're going to end up with things that some of us would call secondary growth, some of us wouldn't. And that's why it's important to identify what's the minimal set of these anatomical fingerprints that can be identified as secondary growth, and we're still working on it with these earliest woody plants. Unsatisfying answer, I know.
Well, thank you.
Mihai, could you look in the chat? There is one question written in the
Yes.
chat.
Okay, I wonder if it's possible to recover, if it was possible to recover fossil plant DNA. Oh, if it was from Devonian sediments they will still conflict with the data on the anatomy of the Devonian fossil plants. I mean, I don't think they would conflict necessarily. That depends on what you're looking at. If you're looking at the regulation of development, we see among major groups of living plants, we see some shared patterns of developmental regulation. So I would expect that I would see the same types of shared patterns, if we had access to Devonian DNA. We also see, for example, if you look, I've been looking recently at leaf development what the regulation of leaf development. And if you look at just angiosperms, there are many fine differences in gene expression patterns between the major transcription factors that determine leaf development within angiosperms. And yet there are still some basic shared patterns that go across all the angiosperms. So I would expect some shared patterns and some differences. But that would be, to me, that's very interesting. But the most interesting aspect of that would be to apply those genes to phylogenetics and include fossils in phylogenetics with the genes. That would be amazing.
Thank you.
Anybody else to ask questions?
Yes, please. I would like to ask the question. Marina Romanova, St. Petersburg University. Thank you very much for this interesting talk. And as an anatomist, I'm excited by the beautiful images and sections of the extinct plants. But also as an anatomist, I'm used to think that all the tissues, including the provascular tissues that give rise to the vascular tissues, differentiate from the shoot apical meristem. So my question is, we know that in plants from diverse lineages, C3HD zips also mark the shoot apical meristems. So do you think that those sterilization of presporogenous tissue involved also the origin of the shoot apical growth in the way of the origin of the premeristem or the shoot apical meristem, as well as it involved the origin of the vascular tissues? Thank you.
That's a great question. I didn't think about that connection. Yeah, it would make sense. I agree. Yeah.
Thank you.
Well, next question. Well, I have my own question. Perhaps not very clever one, because I'm not an anatomist and not a genetic. And so the question related to your first example. Sporogenous tissue sterilized and became a conductive tissue, and it is coded by the same gene. Does it mean that this gene contains the information about the conducting tissue before it existed?
Well, it does not mean that. And so what I'm proposing is that there's a few things here. In living plants, HD-ZIP3 genes diversified. There were duplications, separate duplications in different lineages, and those are shown by the studies of Vasco et al. and Floyd et al. But so what we're talking about is the earliest HD-ZIP, or the earliest function of the HD-ZIP. So what I'm proposing is that they did not have a function in specifying vascular tissue identity. What I'm proposing is that their ancestral function was in specifying pre-sporogenous tissue. So you have a sporophyte, that's a blob of cells, in the earliest polysporangia, in the earliest embryophyte. Within that blob of cells, some are going to undergo meiosis and produce spores. And so that's the sporogenous tissue. So there was some gene that specified the identity of that tissue. But what I'm proposing was that there was another gene, that the HD-ZIP gene, the ancestral HD-ZIP, specified that tissue as potential sporogenous tissue, not exactly sporogenous tissue. And that in order from that pre-sporogenous tissue to become sporogenous tissue, another gene was activated by the ancestral HD-ZIP. Okay, so that's one way. So that's the role of the ancestral HD-ZIP in sporogenous tissue identity. In the same way, that gene in other lineages, in later lineages, activated a different gene that was responsible for that tissue, the pre-sporogenous tissue, become differentiating as vascular tissue. So basically, and that's what I mean by a shared identity. The vascular and sporogenous tissue share an identity in their precursor tissue. So there's this hypothetical tissue that is designated as having the potential to become either sporogenous tissue by the expression of certain genes or vascular tissue by the expression of other genes. But its identity, this potential, this double potential, is specified by the ancestral HD-ZIP. Does that make sense?
Yeah, thanks a lot. Well,
Hello.
where else?
I
Vasilisa?
have a question.
Hi.
Thank you for your presentation. It was really informative. So I want to ask a question closer, maybe to genetic. I don't know. When you talked about the origin of conductive tissue, there was a question in the bryophytes. And as I know, we don't know the exact phylogenetic relationships between the early higher plants. So is there a possibility that bryophytes separated from all the rest very early, and that the conductive tissue arose independently and have different genetic mechanisms from others? Or we can definitely say that bryophytes have the same mechanisms and genes that just for forming conductive tissue.
Oh, that's a great question, and we don't know, right? Because we don't know what the genes that are involved in specifying conducting tissues in the vascular plants. We don't know much about what they do in bryophytes, including HD-ZIP3 genes and others. So the current thinking is, I'm talking about a paper that I contributed to by the people in the developmental labs in the Netherlands, in Belgium, is that that conducting tissues in a very broad sense, in terms of having somewhat specialized cells that for conducting water and conducting food, or photosynthase, they are ancestral to all embryophytes. Well, all embryo. They arose in a common ancestor of bryophytes and tracheophytes, but in a very basic form. And then that was elaborated separately in the bryophytes and in the vascular plants. But that's just a hypothesis, because based on the little that is known about the genes involved in specifying conducting tissues in bryophytes, and based on the taxonomic the presence of of some form of specialized conducting tissues in different taxonomic groups, even among bryophytes. But obviously, the specialized conducting tissues of some sort are not known in all the bryophytes. Only, some mosses and some liverworts have them. And so if you look at them just like that, you can say, well, they are independently evolved. And so it's an example of homoplasy. But the argument we're making in that paper was that we haven't looked carefully enough at bryophytes and at all of the genes that we know are involved in vascular tissue differentiation in other groups, and to look in bryophytes whether they exist and what they do there, and how much what those genes do in bryophytes is related to some form of conducting tissue identity. So we don't know. The answer is we don't know. But it is possible that conducting tissues, in the broadest sense, had a single origin.
Okay, thank you.
Thank you. More questions?
So, good evening, or I don't know which time with you. So my name is Alexei, and I would like to ask you about the plants that cambia without anticlinal divisions. You consider it as a special trend. I looked at your paper with these diagrams, and I had some, and you show some examples of these stems from fossil plants, but in fact, this is quite narrow rings of xylem,
Right.
showing no anticlinal divisions. So my question is honestly saying, these examples are not very convincing for me. Maybe it is just narrow rings. We can observe it in young juvenile stems. And do you know any evidence of constraint for anticlinal divisions in cambia? Maybe genetic or physiological, I don't know. This is my first question.
Okay, I'm bracing myself for the following questions. But okay, so to answer your question, the best evidence for lack of anticlinal divisions is in Sphenophyllum. So it's a carboniferous plant, Sphenophyllum, which has radial aligned tracheids in cross section that don't divide anticlinally. And you're right, that can only happen for a certain amount of secondary growth, past which the cambial initials would have to divide anticlinally, or else the cells that they produce, they become bigger and bigger and bigger and bigger until they reach a size that's beyond sort of survivability for a cell. And that's what we see in Sphenophyllum. In Sphenophyllum, as you go outwards toward the outwards from the center of the stems, you see tracheids in a file. You see tracheids that are larger and larger and larger and larger. And when they reach the largest size, secondary growth stops. And there is, since these are fossils, there's no direct developmental evidence for that. But there's pretty good evidence in the sense that Sphenophyllum only reached a certain size. So all the specimens of Sphenophyllum, none of them goes beyond a certain diameter. And that's probably because they were limited by the lack of anticlinal divisions. So they did some secondary growth until they had to stop. And so you wouldn't find a Sphenophyllum that is, 20 centimeters thick. It doesn't exist. All of them are a few centimeters thick at the most. So that's sort of indirect evidence that, or that explains why, how these could grow without anticlinal division. And the answer is they had, if you will, determinate secondary growth. And it makes sense for sphenophyllum because sphenophyllum was a vine type or liana type plant. It didn't grow as a self-supporting plant, as far as we know, or not a large self-supporting plant. So what controls anticlinal divisions, regulatorily speaking, I don't know, and I don't think anyone knows specifically. Well, but my hypothesis is that it is the ability of the cambial, the fusiform initials to have a specialized response to tangential tensional stress. So I think as these fusiform initials grow tangentially, because the cambium expands or increases in perimeter, at some point the tension in the cells triggers some kind of response mechanism, some kind of gene cascade expression that that determines the anticlinal division. But nobody knows what that is, and nobody has tested that.
Okay, thank you very much. I will check the pictures again. But I have another question. So, shown very, very interesting pictures and of the cross sections of Devonian plants. And is there any correlations between the shape of stela of xylem and some pattern of branching or habit of these plants?
And yeah, so here I assume you're not talking about the secondary xylem. You're talking in general about the primary xylem, the actual steel, the primary xylem, right?
Of course.
Yeah. So
sure.
So unfortunately, not all the plants for which we know the anatomy well, do we also know the morphology and their branching patterns. We do. So the general theory is that as the stems become bigger and they develop more, they evolve more complex lateral appendages, whether those are leaves or specialized branching systems of some sort, that basically stretches the stele, if you will, and it changes from a protostele to a siphonostele or an actinostele. And that's the general idea. Then again, we've been just working on an early Devonian plant that is very, very small. The whole stem or axis is maybe 1.5 millimeters across. And yet that stem has an actual pith. It has a pith, which we don't see in plants of that age at all. This would be the first true pith of an early Devonian plant at a very, very, very tiny size. And also this plant, we know that it had pretty simple lateral appendages that were not too big. So, the theory is something with bigger, thicker stems and more complex lateral appendages, sort of triggering or driving the evolution of more complex stellar architectures. But in reality, if you look at the fossil record, we find all sorts of surprising things. And so we're working on that plant. We're going to submit it within half a year or so, hopefully. Then again, there's this paper that came out recently that says that the complexity of the stele of stellar architecture was not driven by this increase in plant size, but by in stem size and complexity of lateral appendages, but by conduction needs. Gar, can you help me with this? I don't remember what the other hypothesis was that they were proposing. So I know you asked me about it.
Yeah, but you didn't give me a good answer.
Okay.
Because I hadn't had time to think about it.
Yeah, I know. I don't
Thank
know
you
the
very
answer
much.
to that. That's why I asked
No,
you.
no, no, but their hypothesis was that there were strictly hydraulic constraints to of, of, of cell interconnectivity, lateral interconnectivity of xylem cells that were driving that. So I looked at that paper. When I looked at it more carefully, and I asked myself around people who know more about hydraulics and so on, that just doesn't stand up. It's an interesting hypothesis and model, but it doesn't stand up to serious scrutiny. So I'm talking about a paper that came out recently in Nature Plants, maybe a month or two ago at the most. And yeah, it proposes that the diversification of stellar architecture was strictly driven by these hydraulic constraints and not by broader morphological constraints in terms of size and complexity of lateral appendages. That's all I can say. Lots of hypotheses.
Thank you.
Mhm. Thank you very much. Very interesting. Thank you.
Colleagues, more questions?
Can you see me? Yeah,
Yeah.
it's
Mikhail.
Hi, Mihai. It's a great presentation, and Devonian images are very stimulating to look for something like that. And my question is, if you would be eager to find pre-conductive tissues, what you would like what you would look at to find tissue which you would call pre-conductive.
Oh, pre-conductive tissue. I would look on it. I would look for cells that are narrower and more elongated than the cells around it. And I think that would be a minimal set of identifying features. And I'm saying that because that's what we see in all the plants that have some form of conducting tissues. Those tissues tend to have cells that do more elongation than growth in width. That I would look for that, and I would look at the center of the axis, whatever that is, whether it's a gametophyte stem or a sporophyte seta. I would always look for them at the center because that is the plesiomorphic, the ancestral organization of plant tissues in general. You have the conducting tissues in the earliest and simplest plants always at the center. So I'll look at the center and I'll look to see whether there is some kind of differentiation in shape and size of the cells.
Thank you.
Consistent.
So to say that in bryophytes, for example, there are some cells which are long and narrow, and they form the border. And somehow these border cells are usually long and narrow. Are they conductive, or could this be another type of conducting?
Maybe, especially if they're at the center of whatever that is. I would hypothesize that and try to test whether they are conductive or not. If they have a central position. Also, I wanted to say that it's great to see you, and that you will soon receive some papers from us to review about fossil bryophytes from the early Cretaceous.
You probably too. I think that this in this year, definitely.
Good.
More questions?
Yeah, one kind of a follow-up on the last question. We were focusing on cell size, which is a product of cell division. So if we're going to look closer to the apex or in the meristematic region, perhaps it'd be best to look for plane of cell division as a precursor to the types of cells that are long and narrow and would lead to conducting tissues.
I agree. If you can see those, yes, yeah. Yeah, that's a good point. Actually, there has been a lot of recent work on planes of cell division and how they lead, for example, from the protonema stage of bryophytes to growing gametophore stems, if you will, the stems with the parts of the gametophytes with stems and leaves and so on. But those are, yeah, they may be hard to follow, but yeah, planes of cell division. Yeah, I agree.
Mikhail?
Yeah.
Well, more questions? Or perhaps remarks or observations? Well, it seems that it was the last one. Well, if so, thank you so much, and we all was happy. We're happy to see you and to listen to your beautiful lecture and to see your presentation.
Thank you very much for the invitation, and thank you so much, especially those of you in Russia, for sticking around. It's late evening there. I appreciate that, and I'm very honored.
Thank you so much.
Thank you. And there are
Thank
many
you.
thanks in the chat written to you.
Thank you very much.
Thank
Thank
to
you.
Natalia.
Yes.
Absolutely, for organizing all this.
Thank you. Thank you.
Bye-bye.
I was happy to
Bye.
see you all. Bye.
Bye, thank you.